Welcome to this comprehensive exploration of the AXL receptor tyrosine kinase, a fascinating molecular player that has captured the attention of cancer researchers and clinicians worldwide. Today we're going to dive deep into the intricate world of cellular signaling, exploring how a single receptor protein can influence the fate of cells in both health and disease, particularly in the complex landscape of cancer biology. The story of AXL is one that intertwines basic molecular biology with cutting-edge therapeutic development, offering hope for patients while simultaneously challenging our understanding of how cancer cells adapt, survive, and resist treatment. Before we delve into the specifics, it's important to understand that AXL doesn't work in isolation, but rather exists as part of an elaborate network of cellular communication systems that govern everything from how cells grow and divide to how they move, respond to their environment, and interact with the immune system. This receptor, named for the Greek word anexelecto meaning uncontrolled, was first identified in patients with chronic myelogenous leukemia, and since its discovery, researchers have uncovered its involvement in numerous cancer types and its potential as both a diagnostic marker and a therapeutic target. What makes AXL particularly intriguing is not that it acts as a primary oncogenic driver, meaning it doesn't typically cause cancer on its own through direct mutations, but rather that it becomes overexpressed in many different types of cancer and contributes to some of the most challenging aspects of cancer treatment, including drug resistance, metastasis, and immune evasion. Throughout this presentation, we'll explore the molecular structure of AXL and how its unique architecture allows it to function, examine how cells normally regulate this receptor and what happens when that regulation goes awry in cancer, investigate the diverse roles AXL plays in tumor biology from promoting cancer cell survival to helping tumors hide from the immune system, discuss the tumor microenvironment and how AXL influences the complex ecosystem surrounding cancer cells, review the current state of therapeutic development targeting AXL, including both preclinical research and ongoing clinical trials, and finally look ahead to future directions in AXL research and what questions remain to be answered. By the end of this exploration, you'll have a thorough understanding of why AXL has become such an important focus in modern oncology research, and why so many pharmaceutical companies and academic institutions are investing resources into developing ways to inhibit its function. So let's begin our journey into the molecular world of AXL, starting with its basic structure and moving through increasingly complex layers of biological function and clinical relevance. The AXL receptor tyrosine kinase belongs to a small family of related proteins known as the TAM family, which takes its name from the three members TYRO3, AXL, and MERTK, each of which shares similar structural features and functional characteristics while also maintaining distinct roles in different cellular contexts. To understand how AXL works, we first need to appreciate its molecular architecture, which is elegantly designed to receive signals from outside the cell and transmit them to the interior, where they can influence cellular behavior. The AXL protein spans the cell membrane with its structure divided into three main regions: an extracellular domain that projects outward from the cell surface like an antenna ready to receive signals, a single transmembrane domain that anchors the protein in the lipid bilayer of the cell membrane, and an intracellular domain that contains the tyrosine kinase enzymatic activity responsible for propagating signals within the cell. The extracellular portion of AXL contains two immunoglobulin-like domains followed by two fibronectin-type 3 domains, creating a complex three-dimensional structure that serves as the binding site for its primary ligand, a protein called growth arrest-specific gene 6, or GAS6. The interaction between AXL and GAS6 is not a simple lock-and-key mechanism, but rather involves a fascinating ternary complex that also requires the presence of phosphatidylserine, a phospholipid that normally resides on the inner leaflet of the cell membrane, but becomes exposed on the outer surface under certain conditions such as apoptosis or cell stress. This requirement for phosphatidylserine adds an additional layer of regulation to AXL signaling, ensuring that the receptor is activated primarily in specific contexts where this lipid is available, and this mechanism has important implications for understanding how AXL functions in the tumor microenvironment, where dying or stress cells may expose phosphatidylserine. When Gas6 bridges between phosphatidylserine-expressing membranes and the AXL receptor, it induces dimerization or clustering of AXL molecules, bringing the intracellular kinase domains into close proximity, and this proximity allows the kinase domains to phosphorylate each other on specific tyrosine residues in a process called autophosphorylation. These phosphorylated tyrosines then serve as docking sites for various intracellular signaling proteins that contain specialized domains capable of recognizing and binding to phosphorylated tyrosines, initiating a cascade of downstream signaling events. The intracellular kinase domain of AXL shares the characteristic structure of tyrosine kinases with an ADP binding pocket that uses the energy from ADP to add phosphate groups to tyrosine residues on target proteins, and this enzymatic activity is tightly regulated under normal circumstances but can become dysregulated in cancer. The specific tyrosine residues that become phosphorylated on AXL itself include several key sites that recruit different signaling adapter proteins, allowing AXL to simultaneously activate multiple downstream pathways, creating a complex signaling network that influences diverse cellular processes. Beyond the primary Gas6 ligand, research has revealed that AXL can be activated through alternative mechanisms, including ligand-independent activation through interactions with other receptor tyrosine kinases, activation by other proteins in the TAM ligand family, such as protein S, which can also bind to AXL, though, with lower affinity than Gas6. And even mechanical forces or changes in the physical properties of the cell membrane can influence AXL activity. Understanding the structural complexity is crucial because it informs therapeutic strategies. Small molecule inhibitors typically target the ADP binding pocket of the kinase domain, while antibody-based therapies might target the extracellular domain to prevent ligand binding or induce receptor internalization and degradation. The three-dimensional structure of AXL has been solved using X-ray crystallography and other structural biology techniques, providing detailed atomic-level information that has proven invaluable for rational drug design efforts. Interestingly, the kinase domain of AXL shares significant sequence similarity with other receptor tyrosine kinases, which presents both opportunities and challenges for drug development. Opportunities because knowledge gained from targeting other kinases can inform AXL inhibitor design, but challenges because achieving specificity and avoiding off-target effects on other kinases requires careful attention to the subtle structural differences that distinguish AXL from its relatives. The regulation of AXL expression and activity occurs at multiple levels, including transcriptional control, where various transcription factors can increase or decrease the production of AXL mRNA. Post-transcriptional regulation through microRNAs that can bind to AXL mRNA and prevent its translation into protein. Post-translational modifications beyond phosphorylation, such as ubiquitination that can mark AXL for degradation, and proteolytic cleavage that can release a soluble form of AXL called SEXL that may have distinct functions or serve as a decoy to sequester ligand. In normal physiology, AXL expression is carefully controlled and typically restricted to certain cell types, including vascular endothelial cells, where it helps regulate vascular development and maintenance, immune cells, where it plays important roles in regulating inflammatory responses and clearing apoptotic cells, and various epithelial and mesenchymal cell types, where it contributes to normal developmental processes. The Gas6 ligand itself is produced by various cell types and is regulated by vitamin K-dependent gamma carboxylation, meaning that vitamin K is required for Gas6 to achieve its fully functional form capable of binding phosphatidylserine and activating AXL with high affinity. In the context of cancer, the normal regulatory mechanisms that keep AXL activity in check often break down, leading to overexpression or hyperactivation of the receptor, and this dysregulation has been documented across a remarkably diverse array of both solid tumors and hematologic malignancies. suggesting that AXL provides some fundamental advantage to cancer cells regardless of their tissue of origin. In acute myeloid leukemia, AXL overexpression has been associated with poor prognosis and reduced survival, and studies have shown that AXL contributes to leukemic cell survival and proliferation while also promoting resistance to chemotherapy drugs commonly used to treat this disease. In non-small cell lung cancer, which represents about 85% of all lung cancers and is a leading cause of cancer death worldwide, AXL expression is frequently elevated, particularly in adenocarcinoma subtypes, and high AXL levels correlate with advanced stage disease, metastasis to distant sites, and resistance to targeted therapies, including EGFR tyrosine kinase inhibitors that have revolutionized treatment for patients whose tumors harbor EGFR mutations. Gastric adenocarcinoma and colorectal cancer, two common gastrointestinal malignancies, also frequently show increased AXL expression, and in these contexts, AXL has been linked to tumor invasion, metastasis to the liver and other distant organs, and poor patient outcomes.